welcome back to The Big Picture. My guest is Dr. Richard Gronostajski, and we're talking about genes and gene technology. Actually, there's another thing. You're director of the Western New York Stem Cell Culture and Analysis Center. Tell me about that.
Yes, so I don't know when your viewers will be seeing this, but today, the day you're interviewing me, is actually called Stem Cell Appreciation Day. And what stem cells are they're the cells in your body that are responsible for the continuous healing that goes on in your body. So for example, if you get a cut, your skin heals. And it heals because there are cells within your body that sort of that can sense the wound, and they can come out and they can heal it. And so a number of organ systems have these stem cells. So for example, your blood cells turn over all the time, and you're getting new blood cells made every day. And so that's due to what are called hematopoietic stem cells, which are cells that make all the blood cells in your body. And so the Stem Cell Center is trying to investigate ways in which we can use stem cells to treat other diseases for which there are not normal stem cell populations. So, and one of those areas is in the brain. So for example, if you have a stroke or if you have a traumatic brain injury, there's very little new brain cells that are produced after such an event. So we're learning to grow cells in plates out of the body, expand them, and then try to put them back into patients to try to alleviate some causes of people who have suffered traumatic brain injuries, or people who might be suffering from diseases such as Alzheimer's disease, or diseases such as multiple sclerosis, where cells in the body are attacked by the immune system. So stem cells have a promise of being able to cure things like a spinal cord injury, Alzheimer's disease, even diabetes, where diabetes is often caused by a loss of a specific cell population in the body. If we can make new cells and put them back into patients, then we could hopefully, at some point in the future, rather than taking an injection of insulin, we can put cells in your body that would sense insulin the way your normal cells should, and produce insulin so that you no longer have to, for example, take insulin injections.
Well, years ago there was a controversy over where you would get stem cells from, embryos or some other place within the body. Where has that gone now? I mean,
right. So for a long time, the only cells that we could use that were representative of the very earliest stages of human development were what are called embryonic stem cells. And those are actually isolated from very early human embryos. These are embryos that are no more than 50 or 60 cells. So no organs have begun to develop, but those are our early human embryos from which these embryonic stem cells have been derived. But about 10 years ago, a Japanese researcher actually discovered a way of taking cells from adult cells and actually reprogramming them back to where they look like and behave like they're these early embryonic cells. And so these are called induced pluripotent stem cells, or usually abbreviated iPSCs. And these cells have the remarkable ability to, you can grow them in culture, and you can convert them into new brain cells, you can convert them into new muscle cells, into new heart cells, into new pancreatic cells for diabetes. And so there's been a tremendous explosion over the last 10 years of learning how to manipulate these cells and grow them, and then be able to eventually transplant them into patients in which those cells are lacking. And so,
do you see in the future an ability to grow from stem cells, grow organs, complete organs for people, like, you know, say if you have liver disease, if you can take somebody's cells, so there would be no rejection problem, could you maybe grow a new organ for that person?
There are investigators actually at UB who are trying to do that right now. The biggest issue is your organs normally develop in utero, inside the womb. And so they develop by starting off small and then growing big. But when you're an adult, we're trying to replace a very big organ now. And so the idea is we're trying to take these cells and we're trying to create, a lot of what we're doing now is creating something that we call organoids rather than organs, which are tiny little organs that perform the same function as the large organ in your body does. And if we can create maybe hundreds or thousands of these smaller organoids, maybe we can then put them back into the body and replace an organ. There are also attempts to try to get internal organs to be able to heal themselves with what we call gene therapies, where we change the genes that are expressed in an organ and get it to actually grow larger and healthier when it's currently failing.
Well, you know, this is all interesting, fascinating, if you will, technology. You know, when it comes to DNA and to the stem cells and all the things that we've been talking about, and it seems like we're on the cusp of several different breakthroughs because of the technology. What's the most exciting thing that you can think of that's on the horizon that we're just toying with right now?
Right. So I think now, right now, there are a number of genetic diseases. For example, sickle cell anemia, which is a disease suffered from largely in the African American population, and by a large number of them. And it's an unusual disease because it's a disease of the blood cells, and right now there are clinical trials going on to take a person's stem cells out, to modify them by putting in a new copy of the globin gene, which is defective in sickle cell anemia, and then putting those back into a patient so that with one treatment you can essentially cure a genetic disease. And so there are a number of genetic diseases now for which gene therapies are being used in clinical trials, and which have been able to actually people who would have died in their early teens from a lack of an immune system are now living long and healthy lives because they've had a defective gene replaced with gene therapy. And that was something that 10 years ago or 20 years ago was considered very far away. I think certainly over the next, there are currently human beings living now who have had their diseases cured by gene therapy. Very few of them, because this is taking place only in clinical trials to see how effective it is. But I would not be surprised if in the next 3 to 5 years, whether diseases like hemophilia, which there are very small populations, there are genetic trials now going on for gene therapy for hemophilia, for sickle cell anemia, for various other blood cell diseases. I think there are going to be more and more people walking around who've been treated with exogenous genes integrated into their cells that are going to be curing their disease. And I think that's going to be happening. It's happening right now in clinical trials. I think it is going to become a standard therapy in the next 5 to 10 years.
Okay, well, I think we've got a couple minutes left now, 3 minutes left. The last thing I want to talk about is aging. There's 2 ways that people that affect people's life expectancy. There's disease, which will shorten your life, or there's your natural process of aging that's built into your cells, if you will.
Sure.
With everything that we've been talking about, where do you see the future of life expectancy going? Because A, we can cure certain diseases, and B, we can get into the gene structure and perhaps find the gene that's responsible for the aging process. Where do you think that's going to go?
I think that over the next 5 years, we're going to find, we're probably not going to be doing gene therapy to extend life, but what we're going to be finding are small molecules that inhibit some of the processes that are inherent in our normal aging. So one of the processes is oxidation. We all breathe air, and it powers our body, oxidation of chemicals. But you know, rapid oxidation is fire, is burning, it's destructive. And so there are studies going on to inhibit, for example, the oxidation process in the body and to try to extend lifespan. It's known, for example, if you restrict caloric intake, if you fast, if you take animals and fast them every other day, so they eat nothing for a day, and then that actually extends their lifespan. That by fasting you can extend the lifespan of laboratory mice, and some people are doing that now routinely. They're fasting as a way of reducing their metabolism and trying to live longer and healthier lives. And I think we're going to be discovering small molecules that can do this, that people can take as a pill that might extend your natural lifespan. Instead of it being 70 or 80 years in good health, that we might live to 90 or 100 or even 110 or 20 years in good health, if we can just find these processes that are part of the normal aging process and find a way of inhibiting them. And I think that there's a lot of exciting research going on now to do that, and I think that that's something that we can, again, look forward to in, I'd say, 10 to 20 years.
Well, and when you live to be 120 years, hopefully you'll still be watching WBBZ-TV. Thank you for watching The Big Picture. My guest, Professor Richard Gronostajski, and the discussion of genes and DNA. Very interesting topic. And I'm sure we're going to have you back because there's a lot more to talk about.
Thank you for having me, Phil. It's been a pleasure. And those of us at UB are always interested in trying to extend our educational opportunities to the general public, and so anything I can do to help your audience with that, I'm happy to do.
Much appreciated. If you want any more information, you can go to UB's website, and there's a lot of information there. Thanks again for watching The Big Picture, and thanks so much for watching WBBZ-TV. That's it for this time. Join us next week on The Big Picture. We'll see you then.